In this video, we're going to look at DNA and the use of DNA as a clock to estimate when the first humans lived. It is at the point now where you have seen the videos about biblical creation in Genesis 1 and have seen that, at least I have concluded, that the biblical creation story, the history in Genesis 1, is the best explanation for life on earth. However, there are other parts of the book of Genesis that are potentially unbelievable to those who have not studied them, and I just want to cover these topics to assure you that indeed, just like there was scientific support for biblical creation, there is scientific support to show that some of these other aspects of the book of Genesis are also true with scientific support. So this video and the three that follow, these last four videos in this area, will enable us to answer look at some aspects of Genesis, such as Adam and Eve. Did they really exist? We will find from DNA clocks that we can estimate when the first Adam and Eve lived, and we'll see whether that's reasonable or unreasonable. We can also get some information about Noah, also from this DNA clock that we'll talk about here shortly. There are other aspects of Genesis, such as people living 1,000-year lifetimes. Is that reasonable or not? And again, I'll show you where that is plausible, reasonable, and within scientific understanding. And also something about where do the different human races come from. Evolution has a difficult time understanding or describing how we could have different human races. It's unlikely that humans would, since there was no plan in evolution, that we would have ended up with 4 or 5 or more different versions of the human race, of the human species existing all at once, but there is an explanation within the scientific world other than evolution. So in any event, this video and the three that follow will cover some of these auxiliary areas and give you some knowledge about the scientific backing for those areas as well. Now, DNA, which is deoxyribonucleic acid, is a complex part of all living beings, and we'll see that is also another complex organ once you see how complicated it is. It's something that under evolution theory could not have evolved slowly over time until it was useful. It's so unbelievably and wonderfully complex that it had to have some other source other than evolution to be in existence. But anyway, let's look at DNA. First of all, it was discovered that it is so-called double helix. It's two strands here that are entwined with each other in a helical sort of an organization. And when that was discovered, it was given the Nobel Prize for the discoverers to have mapped out just what the shape of this DNA was. Now, it has 4 different types of members to the DNA strings. They're shown here as 4 different colors. They're technically given the name nucleotides, but I won't use that anymore. Instead, let's just think of these as colored beads. So there are 4 different colored beads. There's an auburn one, there's a tan one, there's a green one, and there's a yellow one. And DNA consists of sequences of these 4 different colored beads, totaling about 3 billion building blocks within each DNA segment within your body. So 3 billion of these colored beads form a chain, and we'll see that these are subdivided into subchains called chromosomes in a minute. But basically, your DNA consists of 3 billion of these colored beads all tied together in chains and helical chains. And if you look closely at this particular picture that I've drawn here, it's not a random thing in terms of one chain versus the other chain. Indeed, wherever there is a red or auburn bead on one chain, the opposite bead that it is connected to is tan. And similarly, whenever there's a yellow bead on one chain, the one that is connected to is a green. So it's tan to auburn and yellow and green with each other. And in essence, what that means is that these 2 helical strings, though they are different, they are in effect related to one another, and they're either reciprocal or inverse in some sense, but what it really means is that they both contain the same information in a slightly different form. And so these strings of DNA are in particular form, and the ordering of these beads is the thing that determines your characteristics. We all have very similar order, but there are small differences which can lead to different characteristics of different beings. Now, as I said, that DNA is 3 billion units long, but it's divided into 46 separate strings that we'll see shortly that are called chromosomes, and these chromosomes exist in pairs, which again we'll see shortly. A chromosome of each one of these 46 chromosomes contains genes in it, subparts of it. Again, we'll see that in a minute. And there are a total of about 50,000 genes in human DNA. Those 50,000 genes are the plans or the parts that determine specific characteristics of an individual. A gene for hair color, a gene for the shape of the nose, for example. And these are the things that determine the individual characteristics depending on exactly what form those genes take. Now, the most amazing thing to me about DNA, other than its complexity and interrelationship and separation into pairs of chromosomes and all that, is that each and every one of your 100 trillion cells in your body contains an identical set of DNA containing the blueprints to make all of your cells. Your body, if you saw the cell video, has a number of different types of cells: kidney cells, skin cells, liver cells, bone cells. But each one of your cells, no matter what type, contains within it a complete set of DNA to build or make every other cell as well as the cell in which it resides. And that's really an amazing thing, a complete set of blueprints in every one of your 100 trillion cells. If you want to stop this and look at the DNA, do it now. Now, further details about the complexity of DNA is shown in this slide, and you may also want to stop this if you really want to look at in close detail at what's going on here. But when a cell is about to die, the body knows that, the cell knows it, and before it dies, it reproduces and makes a copy of the DNA, which is the building plan for making the new cell. So the first step within the original cell is to make a copy of the DNA. Once this is done, then the two cellular areas separate and the new cell is constructed from the information in the DNA, whereas the old cell containing the original DNA then can die. And let's look at a little bit at what's going on here. Now, this is a very small segment of the original DNA that's in an individual cell that's about ready to die. What it does is it starts to unwind, and it has unwound to this point in effect. So here is two parts of that original DNA that were initially in helical form, but they have unwound to this point, and the remainder here will continue to unwind. As one of the segments, one of the string of beads unwinds, it is met by these beads of the appropriate color and shape. Again, yellow and green, tan and auburn, are met in such a way that we now then form a copy of the DNA. The result is, here is copy 1, here is copy 2 of the DNA, and when this replication process is all done, we end up with two sets of DNA within the cell that is about to die. Now, this proceeds for all 46 chromosomes, for all 23 pairs of chromosomes, until we have a complete copy of the DNA within the cell, so that the cell can separate, a new cell can begin its construction, and the old cell can die. Again, a very complex, unbelievably complex phenomenon that occurs here, and it happens, if you remember from the cell video, relatively often. Your stomach cells every 2 or 3 days die and are replaced, and your bone cells every 20 or 25 years die and are replaced. So it's a fantastic thing that happens, but that is the reason why we can continue to live, as our cells are worn out and they can be reproduced. So let's take a very simple look at the sub-segments of DNA that we call chromosomes. And so a chromosome is a segment of DNA. It's a part of DNA, and it consists of 23 pairs. So the DNA is divided into 23 pairs, or 46 chromosomes, and these pairs of chromosomes that we'll see in a picture that follows here shortly are not identical, but they're similar. They're not exactly the same, but they're similar. So first of all, a chromosome consists of four different generic parts. One is the tail ends, which are telomeres at the beginning and the end, the telomere region here. We'll see later on in a discussion of genetics how, again, someone gained the Nobel Prize for medicine as a result of understanding how important these telomere zones are. But let's look at the other parts of it. We have the genetic material here. We have these dark black areas are the genes, and I've shown just a very few genes. There may be thousands of genes in each chromosome, but here just 1, 2, 3, 4 genes are shown. And that we then, in addition to that, have the promoter regions, the separating regions that separate genes, kind of a buffer zone between genes so that the end and the beginning of a gene can be isolated to do its business. So that's what a chromosome in general is, and again, there are 23 pairs of them, or 46, that in total make up your own particular DNA. And these chromosomes contain characteristics of you, and genes, contain particular characteristics of you, and that is determined exactly what you're going to look like, act like, and be like. Now here is a picture of the 23 pairs of chromosomes that exist for both a male and a female. I view the, not being a geneticist, a biologist, I view these as kind of pairs of dead worms. That's the way they're presented here. And I've drawn these and taken some artistic license in drawing these, but what you should be getting out of looking at these pairs of chromosomes is that, first of all, that the pairs are similar. I've drawn these by drawing one and then flipping it over and changing its color to indicate that they're similar but not identical. You'd have to look rather closely at this. Again, you might want to stop to take a closer look, but if you look more closely at some of these, like right here, this red chromosome and this green chromosome are similar in shape, but the black area here, which is representative of a genetic material, is thicker, and which means to me it's slightly different from that allied chromosome gene that's on this particular chromosome. So the general idea is that these chromosome pairs are similar but not identical. And it is important to notice that females for the 23rd chromosome have two so-called X chromosomes. So again, just like other chromosome pairs, they're similar but not identical. In females, they're two of the X type. In males, they are an X and a Y. So the male is the only one that has a Y chromosome. Now, in brief explanation for what happens at conception is that the female ovum takes, and each ovum contains exactly 23 chromosomes, and not just a random set of these, but one of each pair of chromosomes. For example, a female egg might have this red one, and then this green one, then the blue one, then the red one, etc. But selecting one of each one. And different ova would select different sets, and so therefore, depending upon which egg is ultimately fertilized, it will have a certain fraction of the mother's chromosomes, but not identical. Therefore, all children will not be identical from the mother's perspective. Now, it is important to notice also that the female will always select one of these two X chromosomes. So one of these two chromosomes, along with one of each of these other pairs, will be selected by the female. A similar thing happens in male sperm, in that the male sperm for the male's DNA will select one of each of these pairs of chromosomes, again, at seemingly a random sort of way. and such that an individual sperm will have a relatively unique set of the male's chromosomes, but the male will always select either an X or a Y, and if it happens to select a Y, the child that is conceived will be a male. So the female has no control over the sex of the child, but the male's contribution to the chromosomes can determine that it is either a male, if it's a Y chromosome that sperm has selected, or a female if the sperm has selected an X chromosome. So that's the brief view of these chromosomes, and what is interesting or what is important in terms of use of this as a clock is that when this conception occurs and the female's selected chromosomes and the male's selected chromosomes come together, they mix and mingle and match is the quasi-technical term that is used to describe what happens. But basically, these chromosomes come together at conception and in the early growth of the new new child. The chromosomes kind of bang into each other and alter them, each one of them themselves, and maybe share a little bit of material one side or the other. The net result of that is that a truly unique child is formed with a set of chromosomes and genes that are not exactly identical to either one of the parents or even identical to the original source that came in from the male and the female. They're slightly altered. That is why children look somewhat like their parents but virtually are not identical to them. Now, one particular part of this DNA is very important, and that is this male Y chromosome. This explanation that I just gave you about mingling of the DNA at conception does not apply to only the Y chromosome. The Y chromosome is virtually unchanged over a long expanse of time. About every 100 to several hundred, up to 1,000 generations of males does the Y chromosome alter and change, and that will be what we'll see as useful as a clock. If you remember back from the cells video, there is a part of DNA outside of these chromosomes which is called mitochondrial DNA. It's outside of the nucleus where these chromosomes preside, and it is that type of DNA, the mitochondrial DNA, that does not also does not change with time. So in any event, Y chromosomes from males and mitochondrial So, for purposes of a clock, we can take samples of male and female DNA in a population, get a group of people and take their samples of their DNA, and when we select the Y male chromosome, it is passed only from male to male. There are no females with Y chromosomes, it's only males. And as I said a few minutes ago, Y male chromosomes do not change very much. They change very slightly from generation to generation. In fact, hundreds of generations before there is any alteration. Similarly, female mitochondrial DNA is passed from the mother to all children, but it is also the type of DNA that does not change very much. Only hundreds or up to 500 to 1,000 generations does the mitochondrial DNA alter. So they both rarely mutate on this mingling process. They rarely mutate at all, although all the other chromosomes are mingling and changing slightly at conception. And fortunately, we know the rate of mutation. We know that from some studies of closed societies, we can estimate, and there have been estimations made of the rate at which both these two types of DNA, Y chromosomes and mitochondrial DNA, mutate, so therefore we can have that as a clock. Now there have been studies done, and actually in the next video you'll see more detail, but this is just one representative study of the use of mitochondrial DNA, in this case, to get an estimate of when the first female lived. So the idea is to first of all get DNA samples from a group of either males or females. This particular data set here is for females. And so in this case, I think it was 145 placenta from females, along with 2 cell lines, were obtained as a source of mitochondrial DNA, and that DNA was studied amongst this group of 147 people from a spread of different races and different locations on Earth. And this DNA was measured to see how much variation there was person to person. Now, if there was a huge variation in the mitochondrial DNA, then that would indicate that there's been a very long time for these mutations to accumulate. Once a mutation occurs for one female, that mutation is passed on to, say, let's next 100 or 200 generations along the line, and then there's maybe another mutation, and that passed on for another 300 or 400 generations. So if there is a large number of variations in this population that you study, then it has been around a long time in order to accumulate all of those changes. On the other hand, if there's been very little variation in this case in the mitochondrial DNA, then that population has been around a very short time and has had not enough time to accumulate all these mutations for the mitochondrial DNA. So what we do after measuring this amount of variation of the DNA, then we use these results that have been found from these studies of closed populations, and it's in the range of 2% to 4% per million years that the mitochondrial DNA and really the Y chromosomes vary with time. And so that in essence becomes a clock, and what we're looking at here is an eyeball that's looking back in time. So that eyeball is right in the present age here is 0 and is looking back in time to see when that first female in this case lived. And with the 2% per million years rule and the 4% million year range, one can look back and see that it looks like with the 4% number that about 65,000 years ago that first female lived, or 125,000 years ago that first female lived. That first female being the one with the original unchanged, unmutated, unadulterated mitochondrial DNA. The average of these works out somewhere, about 90,000 years or so ago. And again, we'll see a more complete set of data here to indicate when the first female and when the first male lived in the next video. But this is just giving you the information about what DNA is, how complex it is, and how DNA can be used as a clock to estimate when the first male and female human being lived. Now, in summary here, we can see that you've already heard me say several times that DNA is a complex organ. It's a complex part of a living being that, by Darwin's definition, would negate or disprove evolution. And it's so wonderfully complex and unbelievably complex that I think it qualifies as a complex organ disproving evolution, or evolution will need to be modified to include it. Now, you've also seen that genetic variations in female mitochondrial DNA can enable us to estimate when the first female lived, to get back to the most recent common ancestor, is the terminology used in genetics. The most recent common ancestor for females, when did she live? And we'll see an estimation of that in the next video. Similarly, we can do exactly the same thing for male Y chromosomes to find the most recent common ancestor for males by taking Y chromosomes from males, measuring how much they are different, and from that difference use it as a clock to estimate when the first male was created. Thank you for watching this. Hope you'll look at the next 2 or 3 videos to finish off this series.